am Deputy BSGS Manager at Dawn of Bioinformatics Ltd.
And alongside, I am Priyam Boshu, the coordinator of this program. We are honored to have you join us for today's session. Now let us begin with a simple picture. Now imagine a patient waits in a hospital bed, hoping for a medicine that works faster. Doctors try, even researchers try, but drug development is very slow, expensive, and uncertain. Now imagine a different scene: a laptop, a protein on screen, and a researcher running simulations instead of mixing chemicals. In minutes, thousands of molecules are tested digitally. This is computer-aided drug design. Today, we explore how a computer can help design drugs and how this technology is transforming the future of medicine.
Now, before we dive deeper into today's scientific journey, let's take a moment to introduce the platform that made this event possible. Bioinformatics is no longer a thing of the future, it's the now. And everything you need to grow in this field from beginner to advanced level is gathered in one place, IBAI, Institute of Bioinformatics and Artificial Intelligence, the host of this event. Here we offer 20 plus beginner friendly short courses, 2 to 4 month project based training programs, structured internships, level 1, level 2, level 3. This is support for wet lab and dry lab bioinformatics, accessible both online and offline for guidance. IBAI offers lifelong access to learning materials, allowing you to learn anytime and grow at your own pace. Our today's event is powered by Dawn of Bioinformatics Ltd, the first bioinformatics-based limited company in Bangladesh. We provide 40 plus advanced services across genomics, proteomics, metagenomics, etc. And alongside, we offer 15 plus professional training programs. Now comes Dawn Lab, where ideas turn into research. Our in-house research facility, Dawn Lab, inspired by Thomas Edison's Menlo Park, is the innovation engine of our ecosystem. It is built on the principle informatics and intelligence is equals to invention. To understand how everything connects, here's the backbone of our ecosystem. IBAI teaches, DOB applies, Dawn Lab innovates. 3 pillars, 1 unified mission: to make Bangladesh a global contributor in the field of bioinformatics and computational biology. We are currently operating from Dhaka and Chattogram, and all details, courses, services, research are just one click away. Visit our website, join us, grow with us. And now, it is our pleasure to welcome our distinguished international guest for this month. Ladies and gentlemen, please join me in honoring Dr. Abhishekh Kumar Verma, PhD in Structural Biochemistry and Bioinformatics, Assistant Professor and Head, Department of Zoology, Monesh Adivasi University, Rajasthan, India.
Dr. Verma's expertise lies in structural biochemistry, computational drug design, protein ligand interactions, and in silico therapeutics development. His research highlights how computer aided drug design can accelerate drug discovery, reduce experimental trial time, and make medical innovation more accessible, even without a $1 million wet lab setup. In a world where drug discovery takes years, his work reminds us that a laptop today can be a mini drug discovery lab.
Dear participants, please join me in welcoming our respected speaker. Dr. Abhishekh Kumar Verma, sir. Our topic today is how a laptop can help cure disease, the power of computer aided drug design. Sir, it is an honor to have you with us. The floor is now yours.
Thank you, Fran, and thank you, Priyam, for the introduction of my here. Okay, so good evening to everyone. Okay, I am a Dr. Abhishekh Kumar Verma, as a working as assistant professor and in charge of the head of Department of Zoology at the Jaiminese Adivasi University, which are located in the Ranpur Kota, Rajasthan, India. Okay, so it is a privilege to be part of the DawnX Sphere series by the Dawn of Bioinformatics Ltd and IBAI Institute. Today, I will share how a laptop, yes, just a laptop, can help us to cure disease using the computer-aided drug designs. Okay, so I will share you my slides. Okay, present this now. And schemes. Okay, so I think in my slide it is visible.
Yes, sir,
Yes,
it's
sir.
Yes,
visible.
sir.
Visible my slides?
जी
Yes, sir.
सर
Yes,
आपका
sir.
स्लाइड शो
Yeah.
हो रहा
Okay.
है
Thank you.
यस सर
Thank you. Thank you. Okay. So today we will learn, okay, and about the how a laptops can help to cure disease. Okay. So this is the power of the computer aided drug designs. Now my slide is no more. What a problem? Okay, that's fine. Okay. So what is the principles of the computer aided drug designs? Okay. So from the CAD. So CAD is the computational revolutions that complements traditional lab-based drug discovery. Like we are divided into two categories. First one is the structure-based drug designs, and second one is a ligand-based drug design. So structure-based drug designs, okay, we are uses the 3D structure protein data. While in the ligand-based drug designs, okay, we are only focused on the chemical features of the ligands. Okay, and then we are performing the molecular dockings. Okay, so molecular docking is the predicts the prepared orientation of the ligands to the particular receptors and estimating the binding affinity. And fourth one is the pharmacophore modeling. If you are we have the like ligands already reported inhibitors, okay, we are identify the essential features which are responsible for the biological activity. And fifth point is the molecular dynamics simulation. So dynamic simulation bring the atomic behavior to virtual experiments. Okay, we are evaluate the stability based on the RMSD analysis, confirmation changes based on the principal component analysis, and the interaction energy, and based on the amino acids which are interact with the atoms of the ligands in the real time atmospheric details. Okay, now what is the emergence of the CAD in the field of the tuberculosis drug disease? We are learns okay how to techniques okay which are useful for the designing of the which types of the inhibitors against. Okay, so today we are discuss about the particular disease. Okay, tuberculosis disease. Okay, how to helpful CAD for the designing of the inhibitors against the Mycobacterium tuberculosis bacteria. Okay, so traditional drug discovery was slow, expensive, and full of the trial and error based. And CAT brings the logic, speed, and precision instead of the testing of the millions of the compounds randomly. And we now test thousands intelligently. Okay, if you see this slides, okay, we are compared to okay how two traditional methods of the drug development and computer aided design is compare. Okay, so first one is like it involves the more trial and the error process in the traditional bed, but we are using the computer aided designs, so it is a more logical see. Okay, and in the traditional method we are involved like blind screening. Okay, we are just pick up the molecules like, and we will perform the wet labs. Okay, then we don't know okay which molecules is the best against the particular drug targets. Okay, but in the CAD it is a specific and mostly target based. Okay, in the traditional methods, it is a more expensive approach to drug development. We need to like $1,000, like in the form dollars. Okay, we need to like lots of the money for the designing of the drug developments. Okay, but in the if we apply the CAD techniques, okay, so it's minimize the cost of the drug developments. So it is a very relatively more laborious and time consuming in the traditional, but CAD it reduces the duration of required in the development of the new drugs. Okay, so in the traditional methods, it involves the sequential steps, but in the CAD, it's the initial steps that are only sequentially, but are the also parallel and the straight forwards. So it involves the separate interdisciplinary drugs development with the more difficult process in the traditional, but in the CAD, if we apply, so it coordinates interdisciplinary drug development with the easier process. Okay, if you see this flow charts, okay, how to divide the computer aided drug designs with the two strategies. The first one is a structure based drug designs. Okay, if you have the structure of the protein ligand complex, okay, so firstly we are identify the binding sites. Okay, then we have performed the dockings, then we have calculate the binding energy, and finally we have performed the molecular dynamic simulations. Okay, then pharmacophore modeling. But in the ligand based drug designs, okay, so we are just use the like different type techniques, QSAR, okay, and pharmacophore modeling. Then we have performed similar patterns, virtual scanning, selection of the compounds, lead optimization, and we have proposed the new inhibitors. Okay, now, okay, the to the CAD. Okay, so CAD is follow the rational pipelines. Okay, from the target identification to the admit optimizations. So each step save the resources while improving the hit quality. Okay, so if you see these slides, okay, so they are the first one is a target identification. Okay, so we are like firstly we are find out the target. Again, they like particular types of the disease. Okay, and like pathways, okay, signaling path, enzymes which are involved in the like different process in the particular organism. Then second one is the structural retrieval. Okay, if you get the knowledge about the target identification, okay, so how to retrieve the structures of the proteins from the database, okay, and how to prepare the proteins for the uh, for techniques of the computer drug designs, okay, like molecular dockings. Then if you are structures prepared, okay, then we are performing the virtual scannings. Okay, if we already have the like database, then we are performing the dockings, then we are performing dynamic simulations, and we are calculating the energy. Then we are also apply the like admit, okay, absorption, distribution, metabolism, excretion, and toxicity. And finally, we are propose the particular types of the leads. Okay, so this is all out of the computer drug design workflow. Okay, but if you have the knowledge, okay, so you also propose and you also hypothesis of the your own computer drug design workflow. If you knowledge, then so molecular dockings, this is the core of the computer drug designs. So first ones, to docking predicts how a molecule fits into the binding sites of the proteins, just like a key in a lock. Tools like Autodock, Glide, Gold, MZ Jot, okay, these are the calculate the binding affinity of the ligands within the protein site, and also guiding us to promising its. Like example is talking workflow proteins. The first one is a if you have the proteins, so firstly we are removing the water molecules, adding the hydrogens, and optimizing the structure by using the software. And second one is a grid generation. Okay, if you we have the like binding sites of the proteins. Okay, so we are decided. Okay, our molecules where is bind. So the in the CAD systems, okay, we are uh, calling the grid generate. So in the grid generation, we are defining the active sites for the docking simulations. Then what is a docking simulation? So we are using the like different algorithm like the Autodock softwares, Glide, Gold to explore the conformation of the ligands, okay, within the binding sites of the proteins, and we are calculate the binding energy and predict the also best finding fitting pose based on the interaction pattern of the amino acids within the proteins. What is the applications of the molecular dockings? The first one is hit identifications. Okay, so we are screening the large compound libraries for the find out the potential inhibitors. Then we are lead optimization. So we are modify the lead compounds for the strong binding and selectivity based on the CAD workflow. And then here we have mechanistic insights. We are also visualize the molecular interaction patterns like the H bonds, hydrophobic contents, pi stacking, electrostatics. These are the types of the interaction, okay, which are seen in the uh, molecular docking patterns. Now this is the applications. Okay, so then we are learn, okay, and explore, okay, and explore how to create which are useful for the designing of the particular types of the inhibitors based on the CAD. Okay, so just we are learns based on the case study which are published in the good high impacting journals. This study. So first case study. The case study first is a they are the strategy for the virtual screening of the natural alkaloids compound which are targeting the NAD enzymes in the mycobacterium tuberculosis. So what is a mycobacterium tuberculosis and what is tuberculosis disease? So tuberculosis is a life-threatening infections which are disease caused by the mycobacterium tuberculosis. This is primarily affecting the lungs and transmitted through the droplets and airborne droplets. Okay, so this is preventable, curable. So TB remains a major global public health problems. This is a very health challenges. And TB remains one of the leading infection killers within the 10.7 million new cases, 1.3 million deaths globally in the according to in the 2024, according to WHO report 2025. And if you see the case of the in the India, so India is continues to carry the world highest TB burden, contributing to 25% of the global TB cases, meaning 2 deaths occurs every 3 minutes in the country. But in the 2019, the COVID-19 pandemic significantly disrupted the TB diagnosis and treatments. So these are causing the increased numbers of the cases between the 2021 to 23. Okay, and there's a slightly recovery begin the 2024. There are some classical symptoms of the active pulmonary TB, which are include the chronic coughs, okay, blood thinning symptoms, fevers, night sweats, and weight loss, and fatigues and digital clubbings. About the 1/4 of the global population is infected with the latent TB. So it is a high risk of the disease progression in the first 2 years after the infection. But TB is preventable, treatable, and curable, yet global progress remains far below and TB strategy milestone, calling for the innovation in the drug designs, diagnostics, and vaccines. Okay, so if we are have the knowledge of the CAD systems, okay, then we are design the inhibitors for the particular drug pathways in the mycobacterium tuberculosis. Let's see in the next slides. Okay, so how to antibiotic targets of the drug targets in the mycobacterium tuberculosis and how to resistance against the particular drug targets. So this is a figure, okay, show the there are different types of the inhibitors which are bind to the particular sites of the particular proteins in the cell of the mycobacterium tuberculosis. Like I'm giving you one examples like the drug targets in the mycobacterium tuberculosis. The first one is cell wall synthesis inhibitor. The inhibitors is isoniazides, ethambutol, beta-lactams, which are blocked to mycolic acids production and the peptoglycan pathways. Like second category is DNA guided inhibitors, okay, which are involved in the DNA replications. This is enzymes which are involved in the DNA replications. So these are the some inhibitors, fluoroquinolones. This is a category of the drugs. So in this category, there are the some drugs. The first one is moxifloxacin, gatifloxacin, which are disrupt the DNA supercoilings. Third one is the RNA polymerase inhibitors. Okay, so this is rapamycin, which are the good inhibitor drugs, which are blocks to RNA synthesis and is a backbone drug in the TB therapy. There are lots of the drug targets in the mycobacterium tuberculosis. Okay, and they are this have the like different medicines again, the different drug targets. But now in the now all are the antibiotics which are show the resistance against the mycobacterium tuberculosis. So there are some reasons. First one is a target mutations which are prevent the drug bindings. Okay. So second one is the efflux pumps which are find out in the mycobacterium tuberculosis which are actively remove the antibiotics from to inside to the outside, causing the interesting resistance. And third one is the cell wall permeability, which are barriers to reduce the drug entry into the bacillus of the mycobacterium tuberculosis. So, so all are the drug targets. Okay, now the resistance. So we need to explore the find out the different drug targets. You know? Yeah. Wait for 5 minutes. Okay. Okay, so we need to explore the like different drug targets in the microbiota tuberculosis. So, so this is a NAD. As a this is a good enzymes as a promising therapeutics drug target in the microbiota tuberculosis. So this is the pathway of the NAD metabolisms. So NAD biosynthesis pathways. Okay, so there are the some three pathways include the de novo, salvage, and the Westhandler pathway. So NAD biosynthesis is a very essential for the tuberculosis growth and virulence and the persistence. So in this pathway, we are include the de novo, press handlers, and the salvage routes, all converging on the NAD productions. So NAD catalyze the NMN, which are converted to NAD. The first committed steps in the pressure handler pathways, and this enzymes, NAD is enzymes, is essentially validated by the genetic knockdowns and metabolic studies by published which by the Rodinova Edwards in the 2020 14. So metabolic bottlenecks. What is the metabolic bottlenecks? Okay, so all are the products of the de novo and salvage finally enter in the pest handler pathway. So in the pest handler pathway, which are first enzymes, the enzyme is the NAD enzymes, which are converted NMN into the NAD. So this is the first enzymes. Okay, so
Sir, your microphone is off. Can you please turn on your microphone? We can't hear you.
Okay.
Abhishekh, sir.
Yeah, I'm now okay.
Yes, sir. Yes, yes, sir.
Okay, wait. Okay, so I think this is covered. Okay, so, there are some key antibiotic targets and resistance mechanism in the mycobacterium tuberculosis. So there are the different drug targets in the mycobacterium tuberculosis. The first one is cell wall synthesis inhibitors. So there are some types of the inhibitors: isoniazides, ethambutolol, beta-lactams, which are block the mycolic acids and the peptidoglycan pathways. Second category is DNA garage inhibitor. So DNA garage is very this is the enzymes, okay, which are involved in the DNA replications in this bacteria. There are some class of the inhibitors against this. The first one is the fluoroquinolones, the moxifloxacin, gatifloxacin, which are disrupt the DNA supercoilings in the DNA replications by blocking the DNA garage enzymes. Third one is the RNA polymerase inhibitors. So they are the most famous medicine is the rifampicin, which are block the RNA synthesis, and this is a backbone drug in the TB therapy. And there's another is the protein synthesis inhibitors. They are the linozolioids, streptomycin, tetracyclines, which are target the ribosomal machinery. There are the lots of the drug targets in the microbial tuberculosis, but now in the current form, all other drug targets show the resistance against the drugs. How to show the antibiotic resistance mechanism? So first one is the target mutations, which are prevent the drug bindings in the pockets of the proteins. So like mutation in the ROPOB, HG, GAID genes. Second strategy is the flex pumps. So these are the presence in the cells which are actively remove the antibiotics from the inside to the outsides, which are causing the interesting resistance. And third one is the cell wall permeability barriers to reduce the drug entry into the bacillus. So now, so how to select and how to design the inhibitors in the particular drug targets? Okay, so in this study, we have choose the NAD enzymes, okay, which are involved in the NAD biosynthesis. To how to in this pathway they are divided into three categories. First one is de novo, second one is a press handler, and third one is a salvage. So all are the products of the de novo and salvage finally enter in the press handler pathways. In the press handler pathways, they are the first promising enzymes, which is NADE. Okay, so NAD enzymes is converted NMN into the NAD with the presence of the ATP molecules. So enzyme accessibility validated by the genetic knockdowns and metabolic studies. And another is this is a inhibition. If we are inhibit these enzymes, okay, so we are deplete the NAD plus productions, collapsing the ATP generation, and final and the redox balance in the all species of the microbacteriums. And okay, so now promising, this is the promising targets which are supported by the in vitro testings and bactericidal effects on the NAD suppressors. So how to inhibit and how to design the particular types of the inhibitors against these enzymes? So there are some inhibition strategies for these enzymes. So there are two major inhibition strategies have been reported. The first one is the competitive inhibitors. So we are targeting the ATP binding sites of the NAD enzymes and the NMN binding catalytic sites of this enzyme. Second strategy is a non-competitive allosteric inhibitors, which are binding at the diamond interface regions of these proteins. So having this show the two structures. The first one is a PDB-IDG 5 days, which are we are downloaded from to RCSB data banks. So in these structures, this enzyme show in the quaternary structures, okay, and which complex with the NADP products. Second structures, they are the PDB-IDG 6BOE. So this is the structures of the NAD enzymes in complex with the reported inhibitors. So there is this is the inhibitors E9A, which are binds in the diamond interface of these proteins. So having show the okay how to designs and how to target the binding sites of these enzymes. So in this lectures, okay, we are I'm sure the two strategic case study. First one, there where we are target the active sites of the ATP NMNs, ATP bindings, ATP binding sites. And another case study, we are sh- show the, okay, so binding sites of the E9A, okay, where the VR ligands binds, okay. So they are the co-crystallic structure of the MTP NAD with the N2 at inhibitors, which are demonstrate the bindings at the dimer interface, and which are induce the unique dimers of the dimer quaternary structure, which are blocking the enzyme functions without the occupying catalytic pockets. Okay, so this study is published in the 2019, okay, by the Osterman et al. They are the such interference binding inhibitors create the structural distortion. We are preventing the productive assembly of the active sites and the emerging strategy for the selective MTB NAD inhibitions. So these findings we are guided how to rational of the our study and how to explore the ATP and animal binding sites and dimer interface focus to the screen the potent NAD inhibitors by using the CAD technology. Okay. No, so what this is the NAD enzymes that targets in tuberculosis. So this is the metabolic enzyme in the pathway pathways. They are the strictly distinct from the human NMNT enzymes, which are allowing the selective inhibition without the host toxicity. So this is a very also important. So why I choose this enzyme? And NMN adenine transferase has been identified as a promising potential drug targets for the development of the new types of the anti-tuberculosis inhibitors. So this is enzymes catalyze the crucial steps in the biosynthesis of the NAD plus and vital coenzymes for the nucleus cellular process like the DNA repairings, redox balance, etc. So first case study, okay, how to design the workflow of the computer aided drug designs and how to find out the particular types of the inhibitor. Okay, so this is a strategy virtually scanning of the natural alkaloids compounds which are targeting this enzyme. So in this library, I have the 413 compounds. All are the category of the alkaloids. So firstly, we are apply the Lipinski rule of the fives, which are have the like the molecular weight less than the 500s and H bond donor less than the 10s and acceptance less than the 5s, etc. So then we are performing the model lockings. So we are get the top 30 hits. Then we are apply the toxicity, and finally we get the top 10 hits. Top 10 hits. So these are top 10 hits. Sorry for disturbance and before time for time. So we have proposed the top 10 compounds. The first one is stapylopines, biciclin, silodionine, pelastron, cornolonins, canadine, songorin, and protopines. So based on the dynamic simulations, we are calculating the binding energy of the MMP-based methods, molecular interaction patterns before the dockings and after the simulations, and we are calculate the global dynamic analysis like the RMSD, RMSF, RG, SHASA, etc. And also we are calculate the essential dynamic analysis. We are the like the PC analysis, free energy landscape analysis, etc. So why I choose the alkaloid compound library in this study? So first one is alkaloids exhibit the strong antibacterial properties. And alkaloid also interact with the multiple targets which are disrupting the various process essential for the survival and replication of the Mycobacterium tuberculosis. There they have the some show the some synergistic effects. So alkaloids also can enhance the efficiency of the existing TB drugs. The structural diversity of the alkaloids, which are provides a rich source of the novel compounds that can be explored for the TB treatment. So these are the natural origins, maybe reduce the toxicity and side effects with compared to the synthetic drugs. So this study is published in the Computational Biology and Medicine Journal. This is a impact factor is 7.7. Now we are showing the results of this study. So these are the binding score top 30 hits. Then we are compared to all the binding energy with the ATP. We are active the controls. Then we are scanning based on the toxicity analysis. So then we are get the top 10 hits compounds, which are show the MS toxicity no and hepatotoxicity is no. So all are the uh, not showing the toxicity in the humans. So these are the doctor structure of the all 10 compounds for the potential competitive inhibitors against these enzymes. So like the first one is a NAD stalopine complex. So show the binding score is a minus 38.49 kilojoule per mole. If you are compared to our controls, so this is higher than with the controls. These are the all are the protein ligand complex of the NAD with the ligands. Now, so this is a RMSD plots of the all the 10 compounds. Where show the all the compounds show the lower fluctuation with compared to the controls. And we are also calculate the MMPBC binding energy of the last 10 nanocent trajectory of these compounds. So all are the compounds show the good binding energy with compared to the controls. So in this, based on the MMPBC binding energy, we have finally proposed the hydrophotopic molecules for the potential possibly inhibits against these targets. Now, this is the interaction patterns of the NAD enzymes, hydrophotopic complex. So, where you see the ATP and hydrophotopics bind in the same binding pockets, we are showing the superimposed RMSD is the 0.10 angstrom. Now, DOC score is 8.9 kilocalorie mole. And during this 100 nanosecond simulation times, show the higher number of the H bond numbers, like the average number is 4. So this is the interaction patterns of the dot complex after the simulations. So hydrogen bonds show the of the dot complex like threonine 13, phenylalanine 14, serine 42, threonine 107, arginine 141, serine 169, threonine 170. So all are these amino acids show the H bonds of again with the atoms of the hydrophotopies. And after these simulations, okay, they are the maintain the interactions of by these amino acids like the threonine 13, phenylalanine 14, and threonine 107, serine 169, threonine 170 also show the interaction with the NAD enzymes. There are this, also some amines show the hydrophobic interaction. So like threonine 13, chlorine 45, alanine 111. These amino acids show the hydrophobic interaction with the atoms of the hydrophotopic molecules. Now, if you see the trajectory analysis, okay, how to molecular dynamic simulation is useful for the calculate like different parameters of the protein ligand complex. Okay, so here you see like RMSD plots. So hydrophotopies show the lower fluctuation with compared to the ATP molecules. Okay, and contribution energy. No. क्या हुआ? मैं बेटा पढ़ता हूँ ना. हाँ. This is the contribution energy of the amino acids which are interact with the hydrophotopies, and we will show the particular energy contribution of the amino acids. So glycine, like glycine 11, show the minus 0.18 kilojoule per mole energy. Threonine 13 and choline 45, threonine 107, and serine 169. If you see the serine 169, it will show the higher binding energy, which means this amino acids tightly interact with the atoms of the hydrophotopic complex. And we also calculate the RMSF, okay, so of this complex. So after the binding of the hydrophotopies, which are show the lower fluctuation of the particular amino acids which are interacting with the hydrophotopic molecules. Now, second case study. Okay, so in this study, we are using the computer-aided drug design strategy like pharmacophore modeling. So, what is the pharmacophore modeling? Pharmacophore, which means they are the we collect and choose the particular features of the molecules, and we design the particular types of the molecules against the exploring of the different drug databases. Okay, so pharmacophore modeling is a divided into two categories. First one is ligand based, which are used only ligands, but second one is a structure based. So we are include the protein ligand complex. So in this case, okay, we have proposed both the strategy, and this study is published in the molecular simulation journals in the Taylor Francis publisher. This is a very good and high impact journals. Okay, like in the first study, ligand based pharmacophore modelings. So first one is ligand based pharmacophore modelings. So we are perform the virtual scanning against the zinc database. In the zinc database, come on, come on. They are performed virtual scanning against the zinc database. So in the zinc database, there are 23.1 million compounds is included. So we are applying the like two protocols. First one is the user protocols. Second one is the user CRT protocols. And based on these protocols, okay, we are get it only 200 hits from to zinc database. And these hits, okay, we are used for the multiple looking study, and then we are performing the model docking against this NAD enzymes. We are calculate and compare all the binding energy with the control E9A, which are reported in the inhibitors. Finally, we are get the 10 hits from the user protocols and 10 hits from to the user CT protocol. Then we are perform the molecular dynamic simulation for the 200 nanoseconds, and we are calculate the RMSD value of the all the complex, and finally we are get the only one hits, okay, based on the dynamic simulation. So this is the best pharmacophore, best inhibitors against the NAD enzymes. Names, the ID is zinc 34698873. Now, second strategy. So in the second strategy, we are applying the structure-based pharmacophore modelings. So we are using the like Pharmate web servers for the designings of the pharmacophores against these targets. So firstly, we are perform the virtual scanning against the different drug databases. Okay, the first one like is a FDA approved and pharmacophore library. Second one is a target diversity library. Third one is a medicinal chemistry library, and fourth one is a Pumpkin databases. So all are the library have the like different numbers of the compounds and the different category of the compounds. Then we are applying the Lipinski rule of the fives in the Pharmate web servers. So finally, we are get the 6 hits from the FDA approved library, 36 hits from to targeted diversity library, and 44 hits to medicinal chemistry library, and 10 hits from the database. Then we have performed the dockings. We are compared to binding energy with the controls, and finally we are get the 1 hits from to 6 apart, 4 hits from to 36, and 4 hits from to 44 hits, and 3 hits from to the 10 hits. Then we have performed the dynamic simulation for the 200 nanoseconds. Okay. Then we have calculated the RMSD, and finally we have proposed the one compounds for the possible inhibitors against the NAD enzymes. So the ID is CCG 30319. So why I show this case study for the this how to design and how to develop the your own protocols for the CADE, and how to screen the different large database, and we are get the like particular inhibitors interest of the ours. So this is the strategy, okay, workflow. We are proposed. This is one of the world strategy proposed, and finally we are get this inhibitors, and then finally published in these journals. So these are the novel types of the workflow in the CADE systems. Let's we are discuss and learn about the results of these two inhibitors. Now we are moving the next slides. So this is a structure of the NAD enzymes. Okay, Vergil E9 inhibitors bind the diamond interface. So these are the amino acids, okay, which are interact with the ligands. So, and another is the ligands. Okay, we are showing the features of the pharmacophore modeling, like aromatic 1, hydrophobic 3, hydrogen acceptor 1, hydrophobic 1, aromatic 2. So these are the these are the pharmacophore features of the E9 inhibitors. And then based on these pharmacophore features, okay, we are explored like different libraries, okay, and we are get the number of the compounds. So the based on the structure-based pharmacomodeling, we are get the CCG 30319 based inhibitors. The binding scores of these molecules is the minus 28.45 kilojoule per moles, which are higher than with the controls. Okay, and for the based on the ligand-wise pharmacomodeling, we get the zinc 34698873 molecules. So binding score of these molecules is minus 33.05 kilojoule per moles, which are higher than with the controls. This is a monodynamic similar results of the proposed inhibitors against the NAD enzymes. If you see the structure-based pharmacophore modelings, so the CCG 30319 show the average number of H bonds in the 5 within the 14 nanoseconds simulation times. As the C alpha RMSD also show the CCG 30319 show the lower fluctuation, which means this molecules is highly stable in the binding pockets of the NAD enzymes. And we also calculate the ligand RMSD to see the ligand RMSD plots. So CCG 30319 show the lower fluctuation with compared to the controls. Now, so all our conclusion is the RMSD analysis show the stability in the NAD bound CCG 30319 with compared to the controls, and the superimposed structure of this complex show the slight change in the structures within the presence of the inhibitors. And also RMSF analysis, which are show the lower fluctuation of the active residues with the presence of these molecules. Okay, so this is the animations. Okay, we are getting some clipping of the these molecules in the binding pockets of the NAD enzymes. So let's start this video. Okay, so this is the inhibitors. Okay, this inhibitors binds in the dimer interface of the NAD enzymes. So if you see this, they are the lower fluctuation in the binding pockets of these proteins. Okay. Now, another is the ligand-based pharmacophore modeling. They are the same patterns, same results. We are done with this in this strategy. So also ligand RMSD show the equal fluctuation with compared to the E9 molecules, and the superimposed structure of this and these molecules show the slight changes in the structure with the presence of the inhibitors, and also RMSF analysis show the lower fluctuation of the active residues with the presence of the inhibitors. These inhibitors. So this is the same molecules. Okay, so how to fluctuates of these proteins with the presence of these molecules, but all over these molecules show the good potential inhibitors against these enzymes. Now, this is the energy contribution of these two molecules. Okay, so the binding energy of CCG 30319 is the minus 102.30, and the binding energy of the zinc 334369 added 73 is the minus 147.07. And also contribution energy of the amino acids of this in. So if you see the tyrosine 37, which are show the high binding energy, which means tyrosine 137 is tightly engaged with these molecules. Okay, so these are the two case study. We are applying like different workflows of the CAD and how to design the workflows and how to use the like different databases, and how to explore and how to perform the dockings, virtual scanning, and dynamic simulation, and like apply like different filters, and finally we get the like particular types of the inhibitors. Okay, now in these slides, how to AI and machine learnings which are useful in the CAD computer-aided drug designs. The first one is the AI tools like AlphaFold. AlphaFold is a very famous databases which are predict the protein structures within the seconds. Okay, so if we are integrate AI in this strategy, virtual scanning pipelines, so we are possibly we are get like good quality of the inhibitors, and we are also accelerate the identification of targets and the validations. Okay, and second one is deep learning in the dockings. If we are apply the deep learning models in the dockings, so we are get the ranks ligand poses faster than the traditional scoring, and we also calculate the automates QSAR methods and virtual scanning workflows. Third one is generative AI for the molecular designs. So we are using the GN, VAA, and the functional learning models, which are helpful for the designs the new molecules entirely from the scratch. So the key message from the slides, so AI is transforming every stage of the computer-aided drug designs, from to targets to therapeutics, and also future lies in the AI integrated data-driven drug discovery. And what is the computational challenges, okay, in the computer-aided drug designs? So we have faced lots of the challenges like inconsistent data sets, high computation cost, and we gap between the in silico and in vivo validation. Possibly, if you have collaboration between the computational biologist and chemist is very crucial. So, okay, so second one is the computational cost. They are the high cost of the model dynamic simulation. We are simulating the large systems or long trajectory like 100 plus nanoseconds. So we are demands the extensive GPU and CPU resources, and also backup of the all the databases. Second one is the quantum mechanism, QQMM calculation. While precise, this is precise, and they are the computationally heavy and time consuming. Second one is the cloud computing and SPC. So this helps to reduce the computation times, but require the technical expertise and fundings, lots of the fundings. Third one is experimental validation and interdisciplinary collaborations. So in silico versus in vivo. Document results must be validated by the experimentally by using the like different assays or crystallography. Second one is collaborative science. If you are integrate the between the computational biologist, medicinal chemist, and pharmacologist and vitals, so we are get the best results. Now another one is the bridging AI and wet labs. If you are translating the computational prediction into clinically relevant drugs candidates, we are demand to multidisciplinary teamworks. We are need to like more labors. So these are the challenges in the computer drug designs. Now, what is the future directions in the computer-aided drug designs? So, quantum computing will be redefine the precision, and second one is a cryo-electron microscope, which are give the real 3D snapshots of the biomolecules, and also AI and omics integration will make personalized drug design in a reality times. If you see the personalized and medicine drug designs, so we are need to advances in omics like genomics, proteomics, and metabolomics, which are enable the individualized target profiling. And we are also AI-powered CAD models are being trained to design the novel types of the drugs tailored to patient-specific mutations, and marks a transition toward the data-driven personalized therapeutics, the future of reproductive healthcare. Now, what is the careers? Okay, and this is opportunity in the CAD. The first one is the bioinformatics and CAD offer the exciting career in the pharma, academia, and startups. So, if you become a, you also get the like structural bioinformatics, chemoinformatics, computational biology, section pharmacology, data science, in life sciences. Second one is you also get the jobs in the industry, and we also start the opportunity. Sorry for disturbance. Actually, my son is not well. That's why. And AI-driven drug discovery startups, pharma and biotech RD roles, and entrepreneurship veterinaries, academics and collaborative research, interdisciplinary projects, grant writing and funding, mentorship, and skill developments. So first one, I would like thank to Dawn of Bioinformatics Limited company, SK Faisal sir, and IBAI institutes and my university, Gemini's Adivasi University, and I welcome collaborations from students and researchers, and feel free to reach out me via this mail. So this is my email ID. If you like, if you have any ideas and any just thinking, okay, just to contact to me. Okay, I will available for any times. So finally, thank you, thank you, thank you for all the times and attention. Let's continue bridging biology and computers to the accelerate human health innovations. So I think today lecture is very useful for all the participants. So let's if you have any questions, okay, any ideas, any just to ask to me, okay, I will be available.
If you want for a Q&A session, if you have time, if you permit us, then we can just pick up one, two participants want to ask questions.
Yeah, okay.
So participants, questions, then you can ask in the comment section. We can pick up one or two more questions from our guest speaker. As you know, given time very as his son is not so well, so we'll take much time. There is a question that Sajid has asked: should we consider FT? We
Yes.
consider the calculation for ADD.
Yes, sure. You also consider DFT, okay, density friction theory in the CAD systems. Okay, why DFT? Okay, the DFT is very important, and how to predict of the how to predict the affinity. Okay, that like the which molecule is best, which molecule is not best. Okay, based on the features of the ligands. So then we are perform the DFT. Okay, by using like different softwares like the Gaussian and etc. It is a software. So DFT is also important. In the case study first, okay, we are also apply the DFT, but we are not showing the results of the DFT, okay, due to the limitation of the times. But we apply, okay. So DFT is a very powerful method, okay, which I included in this. Yeah, any others?
And any other questions from the participants? So another question is that how NGS are related to bioinformatics?
Next generation sequencing is a very now is the very current, it is a very good techniques. Okay, if we know the we don't know the sequence. Okay, but by using the biotechniques, okay, we are analyze the whole sequence of the DNA. Okay, in the particular organisms. So this is a very useful and quick and fast. If you get the information of the whole genome sequencing, okay, whole genome NGS, next generation sequencing, so we are get the information about the targets, mutations, etc. in the particular organisms, and then we are design the like specific types of the inhibitors which are interact with the particular sites of the amino acids, okay, within the proteins. So this is a very good techniques. Okay, so we are also including the CAD systems.
Sir, Sadhan Nazif has elaborated that is molecular dynamic simulation is very much necessary, and if we cannot afford, how can we avoid this? When can we avoid if cannot afford software or do not have necessary software and hardware?
Yeah, actually, molecular docking is only provide the provide the binding affinity. Okay, so within the proteins, but we're not sure now. Okay, we are not sure. Okay, molecules tightly engage with the proteins during the in the long times. That's why we are include the dynamic simulation. dynamic simulation is very important techniques, and it is very useful. So in the particular process and particular times and particular temperature, okay, how to molecule behave, okay, within the proteins. So we are not avoid these techniques, but if you are avoid these techniques, so we are go for the wet labs and confirm that, okay, so this molecule show the good binding affinity against these particular drug targets. So I think is if you have the like limited resources, okay, so we are possibly perform the like simulation for the like 10 nanoseconds, 20 nanoseconds, okay, but this is not enough for the, okay, but now in the current forms, okay, their general requirement is high. Okay, there are some journals asked to like how many times should they perform the simulations, 3 times, 4 times, okay, how to get the like but average frames from to this sequences etc. Okay, so we are not avoid the dynamic simulation, but if we are not have the facility, okay, that's fine, okay, this is okay, but we are not publish the any articles in the good journals based on the only tokens.
Thank you so much, sir, for your valuable time and knowledge. And thank you to all participants for joining us at DawnX Sphere DX-02. We hope today's session inspires you to explore the world where computers meet healthcare innovation. I would like to request all our participants to fill up the feedback form so that we can develop our session further. And your feedback matters for developing our session. And stay connected with IBAI and Dawn of Bioinformatics Ltd. Until next session, take care, stay curious, and keep learning. And good night. I would like to request all our participants to fill up the feedback form so that we can develop the